Good afternoon, everybody, and welcome to episode 53 of our COVID-19 series from the Royal Society of Medicine for health professionals by health professionals. And we are of the moment today because we're talking about vaccines for COVID-19, and we're very lucky to have Maheshi Ramasamy, who is a consultant in infectious diseases from the University of Oxford and is part of the now very well known, shall I say famous, Oxford vaccine team. Mahashi, just tell us a little bit about yourself and a little bit about Andy and Sarah, who you work with in Oxford, and then we'll go on and fire some questions at you.
Thank you for that, Roger. So part of my job is being a consultant here in Oxford at the Oxford University Hospitals, where I treat patients with COVID, amongst other things. And the other part of my job is being a senior clinical researcher at the Oxford Vaccine Group. So the Oxford Vaccine Group is run by Professor Andrew Pollard, who is a paediatrician, who is the chief investigator on the Oxford COVID vaccine trials running across the UK, Brazil, South Africa, and Kenya. I also work with Professor Sarah Gilbert. Now she is the person who designed the Oxford COVID vaccines using an adenovirus, a simian adenovirus vector with an insert encoding the spike protein for SARS-CoV-2.
Great. Well, I think it will be really helpful. We'll talk a lot about the Oxford vaccine because obviously that's where you're involved, but I'd like to talk about the Pfizer BioNTech vaccine and maybe Moderna and perhaps even the Russian and the Chinese vaccines too. But I think, we've heard a lot about vaccines, but not everybody understands how vaccines are created and developed. So tell us a little bit more about how Sarah, whose life work effectively has been creating vaccines, how exactly does the Oxford vaccine work and how was it created?
So we were able to get a bit of a head start on this vaccine really because Sarah had done quite a bit of work using an adenoviral vector called ChAdOx against a different coronavirus, so against the Middle Eastern Respiratory Syndrome coronavirus, MERS, and in fact had created a vaccine against MERS which had been in clinical trials in humans. So that was really the first step to developing the Oxford vaccine. There are a variety of different platforms of COVID vaccines out there, as you've alluded to. So we've used an adenoviral vector-based vaccine, and there are other vaccines that also use this. So for example, the Janssen vaccine, the Russian vaccine, and there are some Chinese vaccines as well that also use adenoviruses, although they all tend to use human adenoviruses rather than chimpanzee adenoviruses, as we do. There are some platforms that use mRNA, so messenger RNA, including the Pfizer BioNTech vaccine and Moderna. And there are some that use inactivated viruses. So these are COVID viruses which have been inactivated, usually using a chemical or a heat mechanism, so they're no longer able to replicate. And there are several Chinese vaccines that are in clinical trials that have used this. And then finally, of the last group of vaccines that's in phase 3 clinical trials, there's also a protein adjuvanted vaccine. So that's Novavax. So that takes a spike protein and uses a sort of kick to the immune system called an adjuvant and uses that as a vaccine. The majority of all SARS-CoV-2 vaccines are based on the spike glycoprotein. So that's the bit of the coronavirus on the outer coating of the coronavirus that is important in cell entry.
Okay, well, we're going to get lots of questions in. I already had about 100 questions before we even started, but I'll just choose one or two. Gerard Leitung is saying, how different is the Oxford vaccine from the Pfizer one? Why the difference in transport temperature? Which I think basically that question is saying, tell us the difference between a DNA-based vaccine and an RNA-based vaccine, and why does the Pfizer vaccine have to be stored at this temperature of -80, which is causing, which makes it a little more difficult to distribute?
So our vaccine is based on a non-replicating virus. So it's an intact virus with the DNA encoding the spike protein sitting inside the virus particle. That's relatively chemically stable as compared to mRNA. RNA is actually chemically quite an unstable molecule, which is why it has to be stored at such low temperatures. So it comes down to the difference between the different viral platforms, vaccine platforms rather.
Right, and tell us a little bit about the production issues, because it's one thing for wonderful Sarah to create a vaccine, but it's a whole different ballgame creating millions of doses. And I suppose that's why Oxford hooked up with AstraZeneca, a company which is part Swedish and part UK, isn't it? And tell us a little bit about the production facilities that have been created in order to do that, and how do you produce millions of doses of a single vaccine?
So that was a real challenge. So in terms of the very early studies that were done in animals to look to see whether the vaccine was immunogenic and safe in animals, those batches of vaccine were made in Oxford. However, the quality of a vaccine that needs to be used in humans is quite different to that used to a vaccine or to any drug that's used in animals. So there are very strict criteria for manufacture of a drug, including vaccines that go into humans, and this is called good manufacturing practice, or GMP standards, and those are very carefully regulated by in the UK by the MHRA and by regulatory authorities around the world. So first off, there was the challenge of creating or of manufacturing the vaccine to GMP standards within the UK, within Oxford, for us to be able to use it in our phase 1 study. And that was done at the Clinical Biomanufacturing Facility, which is part of the University of Oxford, who have experience in synthesizing, creating these vectors for other diseases for use in other clinical trials that we've performed within Oxford against diseases other than COVID. However, to make the next step, to make several thousand doses, that involved quite a complex piece of work, trying to upscale to allow us to be able to create lots of virus particles to be able to make tens of thousands of doses. And there was a huge piece of work done by investigators here in Oxford to work out what the best mechanism of doing that within this very strict regulatory framework of creating good quality drug product that is safe enough to be used in humans. And that's just for the trial, but then with following on from that, then there's even further scale up that needs to be done to create millions and billions of doses. And that's one of the reasons why we partnered with AstraZeneca, because as a university, we just don't have the capacity to create, to manufacture that sort of scale. And we partnered with AstraZeneca, who are able to draw on their considerable resources to do just this, and they have a variety of manufacturing plants around the world who are working, as I understand, on upscaling production of the ChAdOx vaccine.
Great, and you got government funding to help with that, according to the media. Well, obviously you're a clinician and you work on the wards, and you were telling me that you're going back to the wards after you finish talking to us. So thank you for doing that. But the clinical trials obviously are crucial in proving that your vaccine is safe, number one, and effective, number two. So just talk us through the clinical trial process for not only for your vaccine, but for other vaccines. How do you prove that a vaccine is A, safe, and B, effective? What are your thoughts on that?
So clinical trials, all clinical drug trials are very, very carefully regulated. They can't just be done by anybody. They can only be done by accredited, by organizations that have experience in conducting them. So these are often academic institutions, or they are pharmaceutical companies. So not anyone can just run a clinical trial for a drug. When starting, there's often animal safety and immunogenicity data that needs to happen before we can start using a vaccine or any other drug in humans. When you design a study, there's something called a study protocol. So this is like a roadmap of how the clinical trial runs. So this very carefully lays out exactly what you're looking for, how you're going to do it, and lays out in detail what the implication is for your study participants, how many of them you're going to recruit, what they're going to have, how often you're going to see them, what tests you're going to do on them, and then defines your study endpoints. That has to be approved by the medicines regulator for that country, and also by an independent ethics board, to make sure that you're asking the right questions and you're not putting your participants at risk. Once that happens, once that protocol has been approved, you can start your phase 1 study. So this is the very first time a drug or a vaccine has been in humans, and you usually give the drug to a very small number of people. You follow them up very, very carefully, looking for any signs of safety concerns. You would then move on to phase 2, phase 3 studies, where you perhaps might change your dose of the drug, you might change the age group you're looking in, you're looking for clinical outcomes like efficacy in the case of a vaccine. And these are much bigger studies, because in order to be able to generate statistically significant data, you need more participants. But at each stage, each of those trials will have a separate study protocol and will need to be approved, as I said, by the drug regulators and also by by an ethics committee. Most of these trials, all of our trials, have also had an independent data and safety monitoring committee. So this is a separate group of experts, clinicians who have experience in running clinical trials, who analyze the data regularly to make sure that there is no safety signal, and they give an independent, unbiased review of the data.
And the data that you get, you have your control placebo group, and you have your vaccinated group with the live with the actual vaccine. And I suppose your definition of efficacy is the number of people who actually develop COVID. Is that severe COVID, mild COVID, or is it the antibody response to COVID? How do you decide how effective your vaccine is in terms of patients being benefited from not getting the problem?
So that's a good question. Efficacy is defined in slightly different ways, in different trials, and so it's quite difficult to compare between them. So what's critical is your definition, for example, of COVID disease. In our trial, we only considered participants who had virologically confirmed COVID disease, so a PCR positive swab, plus one of either cough, fever above 37.8 degrees, anosmia, so loss of smell, or ageusia, loss of taste. So we had very rigid definitions of what COVID was. You then also have to factor in your time frame of looking for protection or not from the disease. So when after your vaccine are you going to start looking for efficacy? The adaptive immune response that you stimulate with vaccines only starts to respond to a stimulus by about day 7 after it's first met an antigen. And with our vaccine, the Oxford vaccine, we actually have a 2-dose regime. So we only started looking at efficacy at 14 days after our second dose. So so depending on just what criteria you're using, that's how you would define efficacy, clinical efficacy, and we've defined it as protection from those symptoms of disease. There are secondary endpoints you can look at, like prevention of severe disease, death, hospitalization. And in fact, we were able to show that there were no cases of hospitalized COVID in participants who received the ChAdOx vaccine, which is very encouraging. In terms of antibody response, that's a tricky one, because we don't yet know what constitutes a protective antibody response. So, for example, there are some diseases, like, say, meningitis, where we have a correlative protection. We know that if you have a certain degree of functional antibody measured using a specific assay, then you're unlikely to get invasive meningococcal disease. We don't have that yet for COVID. We don't really know what level of antibody equates with protection. What we do know is that there are certain types of functional antibodies, so neutralizing antibodies. So these are antibodies that bind onto the virus and prevent it from entering target cells and prevent we hope therefore prevent primary infection. We know that in animal models, the development of neutralizing antibodies correlates with protection from challenge. So that's what many of the vaccine trials have been looking at, the development of neutralizing antibody, but we don't yet know whether a given titer of neutralizing antibody is associated with protection.
I suppose we don't know how long those antibodies or protection will last yet, because we're in early days. So one of the things that I think has mystified the profession is that we're reading all these press releases, and we're hearing the politicians come on the telly and talking about this vaccine, but none of the papers have actually been published yet. So we haven't been able to look at the real data. I mean, I like your thought about that. Whether is it ethical to tell the public about these things before the facts are out there for to be looked at in the journals, number one. And number two, there's obviously going to be massive competition between Pfizer, Moderna, AstraZeneca, et cetera, et cetera, et cetera. How are we going to sort out, which is the best vaccine to have? So tell us about publications first, and your thoughts about press releases versus publications.
So you're right, we don't normally release academic results by press release, and it's a sort of byproduct of the intense media scrutiny that there has been on all the COVID vaccine trials around the world. And people are desperate for answers, and there's huge amounts of pressure to release results as quickly as possible. With our phase 1-2 and our phase 2-3 studies, we've already published data on that data is freely available. We've published that in the Lancet. In terms of the efficacy press release, and I think this is something that's been the case not only for us, but also for Pfizer, BioNTech, and for Moderna. When you're working with a commercial organization, if there is a piece of information that might affect the share price, then the organization is required to release that within 24 hours of being made aware of that, and that's to prevent insider trading. So there are very strict financial regulations around that, and that is why these preliminary results have been released in the media. That's not how we would normally do it. They're real sort of snapshots of results, and what we really need is good quality peer-reviewed data, so we can present all our results. They've been independently reviewed by academics who agree with this being published, and certainly we are keen to get our data out very soon, and we hope to have something out in press within the next week or two.
Can you clear up the question about the half dose first dose versus the full dose second dose? Because that's been reported in the media. There's a lot of mystification over that. I mean, I guess, is that embargoed information, or can you tell us a little more about that?
So what I can tell you is that we defined our primary analysis endpoint in our statistical analysis plan right at the outset. We agreed that with regulators, and that was to look at a combined efficacy analysis across our trials in the UK, Brazil, and South Africa. And so we reported the results that we said we were going to, which was our overall figure of 70% efficacy. However, as a secondary analysis, when we looked in more detail at that, we saw that there appeared to be higher efficacy in a group that have had low dose, high dose vaccine, as opposed to groups that had the standard dose vaccine. So the standard dose was just over 60%, and the low dose standard dose was about 90%. And that was really interesting, and we don't really understand why that is, but there are a sort of number of things that could be underlying that. One is sort of anti-vector immune responses. So that's something that we need to tease apart. One thing could be the sort of age of different participants, or the interval between the doses. And that's something that we are picking apart in our efficacy analysis at the moment. One of the frustrating things is that although we had the cases and we could report on the efficacy, we didn't have the immunogenicity data for those participants. So we didn't have, when we announced 2 weeks ago, we didn't have, the antibody titers for each of those groups. That's something else that we're looking in detail at going forward. So yes, we hope to publish all of that soon.
You're in a very high pressure situation. I appreciate that. But what about the safety data, Maheshi? I know everybody's going to be worried about the safety in terms of side effects. What side effects have you seen in the AZ trials, and any information about any of the other vaccines in terms of safety?
So what we saw with our vaccine was that it was actually very well tolerated. And we looked at 3 different age groups. We looked at people aged 18 to 55, 56 to 69, and then over 70 years of age. And we saw that across all those age groups, the vaccine was pretty well tolerated. So it had the same sorts of symptoms that you would expect after, say, a flu vaccine. So local symptoms such as pain or tenderness at the injection site, and then some systemic symptoms such as fatigue, low-grade fever, myalgia, all of which resolved within a few days of the vaccine. So there were no unexpected reactions in terms of the vaccine itself. What was also particularly intriguing was that the elderly seemed to actually tolerate the vaccine a bit better than younger people. So we saw less reactogenicity in the over 70s than we did in the 18 to 55-year-old cohort. And what was also interesting was that after the second dose of vaccine, we saw that there was even less reactogenicity. So that's quite encouraging for vaccine uptake. We hope that it's a vaccine that doesn't cause too much in terms of adverse reactions. In terms of the other vaccines, most of the vaccines that are in phase 3 trials have published data on reactogenicity and safety. And they vary from vaccine to vaccine, but there have been no significant safety concerns to my knowledge with the published data that I've seen so far.
I see we've got 81 questions. I promise you we do look at the questions, and we will take a careful consideration of those, but I haven't got time to ask them. But I will ask Xavier de Souza's question about, does the vaccine work less well in patients with autoimmune diseases such as rheumatoid arthritis or other forms of immunocompromise, I suppose?
With the early vaccine trials, the early stage vaccine trials, phase 1, phase 2, we're obliged by the regulators to only recruit healthy people. So we did exclude people with who are immunosuppressed and with chronic comorbidities from those early phase data. So we don't have the data on how effective or how immunogenic the vaccine is in people who are immunosuppressed, but that is something that we're collecting going forward. In terms of safety, the Oxford vaccine and the mRNA vaccines do not cause infection in themselves. So the mRNA vaccine is a bit of a virus. It's part of the genetic code of the virus. It can't cause infection. And our vaccine uses a chimpanzee adenovirus, a harmless cold virus, as the vector to deliver the spike protein stimulus to the immune system. But our viral vector is replication deficient. It means it cannot reproduce in human cells. And that's because we've genetically engineered it to remove some of the genes that are important that are essential for viral replication. So our virus does not replicate. In fact, the only way we can grow it in the manufacturing plant is by using a human cell line that contains this extra gene. It does not grow in normal human cells.
So people can be reassured about that. Let's move on and talk about rollout, Maheshi. I believe that they've got 800,000 doses of the Pfizer vaccine coming in. It's got to be given twice, so that's 400,000 people. But there are about 400,000 people in care homes. And then there's 1 million employees of the NHS and about 300,000 care home employees. I mean, the rollout is going to be a major thing, isn't it? So tell me a little bit about the plans for that and for the future.
So I'm not an expert in the vaccine rollout. I think it's obviously going to be a huge logistical undertaking to get the vaccine out to those high priority groups. And as you've alluded to, the first group of people to get it are individuals living in, adults living in care homes and people who care for them, who work in those care homes, followed by adults over the age of 80 and healthcare workers. And then there's a sort of age de-escalation, and this has all been set by the JCVI, the Joint Committee for Vaccines and Immunisation, who determine the immunisation policy for the UK. I think it's going to take a huge amount of effort and planning to be able to get the vaccines out quickly to those at-risk groups. And the vaccine manufacturers are going to have to work very closely with the local logistics network. So, for example, for the mRNA vaccines that need colder storage, so -70 to -80 storage, that's going to limit the sites at which that vaccine can be distributed and delivered. But I'm sure there are clever people thinking about exactly how to do that.
I suppose you could say that for the Pfizer vaccine, the people have got to come to the vaccine rather than the vaccine going to the people, which is tricky for the care homes. Whilst the AstraZeneca vaccine, will be easier to distribute to GP surgeries and into the care homes too. So when do you think, any idea of when the AstraZeneca vaccine might get through the MHRA regulatory authorities?
We're hopeful that it'll be before the end of this year, but it's very much up to the MHRA as to when they are ready to approve it.
Yeah. And what are your thoughts about, some people are saying it's quite extraordinary that the UK MHRA has approved the Pfizer vaccine before the FDA and before the European regulatory authorities. Any comment on that?
The MHRA are very robust at their investigation of the data. I have confidence in them. If they have approved a vaccine, then that will be because they believe that all the data that they've seen means that it's safe and effective for it to be used.
And that kind of leads on to the question. I mean, there is a massive amount on social media from the opponents of vaccination, anti-vax group, who are quite vocal. So what would you say to people who feel who are very anti? I mean, some people are worried that it's going to become obligatory. Everybody's going to have to be vaccinated because the government say, just like they say that, we're not allowed to go to the pub and we've got to have the rule of 6 and not allowed to mix and so on and so forth. So we're getting all these instructions from the government. Are they going to say, listen, everybody's got to have a vaccine?
I think that would be counterproductive.
Yeah.
I think there have been lots of studies that have looked at whether making vaccines compulsory increase vaccine uptake, and I think they don't. People need to feel that they are making a personal, individualized choice, and we need to respect their autonomy. I think that overall, my understanding of the landscape is that in general, there's people are quite supportive of taking the vaccine, and they understand the importance of it. I think what you've got to do, and this is crucial as vaccine rollout happens, is that you've got to explain the rationale behind the need for vaccination. You've got to present the safety data, the efficacy data, and we've got to be as transparent as we can be, so that people can make an informed choice and decide to take that vaccine or not. And I'm hopeful that people will. I personally will be offered a vaccine next week, and I will be taking whichever vaccine I'm offered.
Well, they say the Prime Minister is going to be vaccinated on television himself, but I'm not sure whether that's going to be a positive or a negative. Well, we better not go there. Well, and other issues around vaccination, if you've got a few more minutes, people are saying there's a risk of cyber espionage, and there's risk that criminals will produce bogus vaccines that you can go and you can get them done privately in a private clinic. What are your thoughts about that?
I think that's real danger going forward. In terms of cyber espionage, the university has been aware of potential attacks in the past, and we're very lucky that we're very well supported in terms of cybersecurity locally. In terms of fake vaccines, I think that again is a huge problem going forward, and I think we've got to be, and that will just massively reduce people's confidence in the vaccine if we can't guarantee that they're getting what they think they're getting. So I think there's a big piece of work to do there. Actually, Roger, I wrote down before a question that I never answered, which was about competition between the different vaccine groups. Did you want me to talk about that? Or
yeah, please, yeah.
I guess it was really just to say that we don't yet know with each of these vaccines, how long protection is going to last, because we haven't got the long-term data on that yet, and we don't know which groups are best protected by the vaccine. But it's really encouraging that we have already, several vaccines that are reporting efficacy. And, this absolutely isn't a vaccine race. We're likely, what we need is lots of vaccines that work, and lots of vaccines that work in different groups of people. And so it's great that there are lots of vaccines that are out there. We're not in a competition. We're all absolutely on the same side.
Well, Karen Butterfield actually sent a question in before this. She said, will patients be able to choose which vaccine they receive? When I go into Waitrose, I can choose what I have for my supper, or which bottle of wine to buy more commonly. Will they be able to choose, do you think, or will...
At the moment, I think it's going to be logistics that affect which vaccine you're offered. For the first part of rollout, clearly it's going to be Pfizer, and later on there might be our vaccine or other vaccines coming along as well. But I think it's important. What's really critical is good vaccine coverage in order to be able to reduce the peak of the epidemic and to prevent people getting sick. What we need is good vaccine coverage. And so you should take the vaccine that you're offered. Next week I'm likely to be offered the Pfizer vaccine. While I would love to have the Oxford vaccine, I'm going to take whichever vaccine I'm offered.
Yeah. That kind of leads me to a question. I can see some young people might say, well, I'm young, I'm fit, I'm unlikely to get COVID seriously. If I get it, it'll be mild, probably won't, maybe asymptomatic. So why should I have a vaccine when I don't need protection? What would you say to somebody that said that to you in your clinic?
It's according to the current JCVI prioritization, young, healthy individuals are likely to be quite late are likely to be offered the vaccine quite late. So they're unlikely to be in the first few tranches of the vaccine rollout. However, even if you individually are not at risk of severe disease, there will be friends and your family members who are more vulnerable than you, who may well be. And so I think, if it comes to the stage where we are offering vaccine to young, healthy people, there might be impacts of the vaccine on transmission, and that would be a game changer in terms of controlling the epidemic. We've all had such a rubbish year, with social distancing and all the personal and financial setbacks we've had this year and restrictions we've had this year. I think if you're offered a vaccine, you've got to weigh up whether playing your part in reducing that and preventing that happening again, whether that's worth it. And I personally think it is.
Absolutely right. I think there was an ambulance behind you, not me, wasn't it? That's the next patient coming into the famous Radcliffe Hospital there, I should think. Well, thank you so much, Maheshi. It's been wonderful to speak to you, and I know that it's very early days yet, and we're really looking forward to reading the scientific literature as it appears, hopefully next week, as you say, and to hearing more about this absolutely fascinating subject. So don't go away. I just need to tell everybody about a few other things. Well, luckily next week, the COVID-19 series continues, and we've got Professor Tim Spector from King's College Hospital, who has some fascinating information about the app that they've developed there, which has given them so much information about the way that the COVID-19 pandemic has spread through the UK and beyond. And he'll be here this time next week, again, talking to me. And the title of his talk will be COVID-19 1 year on, what have we learned? So we're going to go right back to the beginning of this pandemic, and we're going to analyze what's been done, what should have been done, what's been done right, what's been done wrong. And we'll also speculate a little bit about the future as well. Next week, also, Simon Wesley will be back talking to Rachel Clark, very famous, another doctor from Oxford, very famous, great communicator. So she's in conversation with Simon Wesley on Wednesday evening next week. So thank you, everybody, for listening. I'm so sorry I didn't get a chance to put all your questions. As I say, we will take a look at those. Please do remember that the RSM is under the cosh because our building's closed. And unlike Oxford University, which gets billions of pounds of support, poor old RSM has to soldier on through the pandemic. So if you would like to make a contribution to help us continue with our program of education and innovation at the RSM, we would really love to receive that. So thanks again, Maheshi. Please do say thank you also to Andrew and Sarah. I read a most amazing report in the Financial Times weekend about Sarah and the way that she's devoted her whole life to creating this. She must be very proud of what she's done. You all must be very proud. So congratulations, many thanks. And we'll see you next week, everybody. Bye for now.